everyone. My name is Bala. I'm a postdoc in Samantha Bell's lab at Rutgers, New Jersey Medical School. Today, I'm going to talk about host pathogen interactions of Mycobacterium tuberculosis. Before I start, I would like to thank the organizers for giving me this opportunity. We've had an excellent introduction to tuberculosis from Dr. Yang's lab, so I'm going to be brief about how TB is bad. Mycobacterium tuberculosis causes the disease tuberculosis, and it infects 10 million people globally and kills about 1.2 million people annually. In our lab, we are interested in studying host pathogen interactions of Mycobacterium tuberculosis. Macrophages are the first line of defense against invaders, and they're generally efficient in killing them. But MTB prevents this, and it survives and multiplies within macrophages. It does this through secretion of effector proteins, which first poke holes in the phagosomal membrane and get cytosolic access, and then they modulate macrophage cell biology. Among such effectors, several of them interact with host immune factors, and in our lab, we are interested in studying one family of such effectors called PEPPs. This large family of 170 proteins get their name from the conserved proline glutamic acid and proline glutamic acid residues in the N-terminal. And this family is particularly expanded in pathogenic mycobacteria, constituting about 10% of its genome, suggesting that they're all very important for bacterial pathogenesis. However, a large portion of them remain uncharacterized. Today, I'm going to talk about one protein, PE5, from this family, and we are particularly interested in PE5 because it is conserved across several mycobacterial species, including the human pathogen MTB and cattle and fish pathogen Bovis and Marinum. To characterize the function of PE5, we adapted this approach where I cloned PE5 from MTB and expressed it and cloned it with a GFP tag into a mammalian expression vector and expressed this in HEC293 T cells. I then used affinity purification using a GFP trap to identify PE5 interacting proteins using mass spectrometry. When I used this approach, and I also used GFP only cells as the negative control, I identified several interactors of PE5, and some of them formed part of this complex called CRL2 complex. This complex is a modular E3 ligase complex where CUL2 is the E3 ligase, and it recruits the target substrates using these receptors. These receptors identify specific residues in the target protein, and they bring these proteins to the CUL2 for it to ubiquitinate and target it to degradation using proteasome. Among our mass spec hits, we identified KLHTC2, which identifies proteins ending in diglycine, and PE5, which ends in diglycine, makes an ideal substrate. So the first thing I wanted to do was to verify this interaction. To do this, I performed directed co-IPs. In this, I used the GFP tag PE5 with flag tag KLHTC2, and I again performed GFP pull down. And I saw that when I used this, flag tagged KLHTC2 did come down with PE5, suggesting that PE5 and KLHTC2 do interact. Now, to test their interaction a little further, I generated a KLHTC2 mutant which lacks its substrate binding activity. And when I did a co-IP with PE5, the mutant did not come down with PE5, suggesting that PE5 binds at the substrate binding pocket of KLHTC2. Now, to test this further, I generated another mutant, this time a PE5 mutant which lacks the terminal diglycine. And I again saw that this mutant also could not interact with wild type KLHTC2. And this suggests that PE5, through its terminal diglycine, interacts with KLHTC2 at its substrate binding pocket. Now, we were excited about this finding because CRL2 complex has been traditionally studied only in the context of ubiquitin machinery and proteasome and protein homeostasis. But since MTB has a specialized effector protein which interacts and possibly also blocks this complex, we think it can have an important role in bacterial pathogenesis. So we wanted to test this, and to do this, I used this inhibitor MLN4924, which blocks the activity of CRL2 complex. And I infected these cells with MTB. And I saw that when CRL2 complex's activity is inhibited, there is an increased MTB replication in these cells, suggesting that CRL2 is actually important for controlling MTB replication. Now, to test if this is because of KLHDC2, I used shRNA to knock down KLHDC2, and I again infected these cells. And I again saw that even in the knockdown cells, KLHDC2 knockdown cells, there is an increased MTB replication. This suggests that KLHDC2 and CRL2 complex are important for controlling MTB replication. Now, we were very excited because we could identify novel host restriction functions of CRL2 complex. And we wanted to see if this was specific to MTB or if other bacteria are also controlled by this complex. To test that, I used Salmonella as an organism. And I again did the same approach. I used MLN to block the activity of CRL2 complex. And when I infected them, although there is no significance, we saw an increased Salmonella replication in cells lacking CRL2 activity. I also did the same KLHTC2 approach to see if it would promote Salmonella replication, and I saw that this time I even have significance, and there is a greater bacterial replication in KLHDC2 knockdown cells. This was really exciting. So the next thing I wanted to do was PE5 is a MTB protein. Salmonella doesn't have PE5. So I thought if I provided PE5 by ectopically expressing it in macrophages, I wanted to check if that also promotes Salmonella replication. And that's what I've seen, that when I have provided PE5 within macrophages and I infected these macrophages with Salmonella, there is more Salmonella replication in these cells, suggesting that PE5 is doing the activity of blocking CRL2 and KLHDC2, and that's how it is promoting bacterial replication. So the next thing we wanted to do was to identify how this is happening. So I went back to PE5 and I wanted to understand the mechanistic consequences of its interaction with CRL2 complex. So CRL2 complex is a ubiquitin machinery, so it adds ubiquitin onto the target substrates, and these ubiquitinated proteins are then sent to proteasome for degradation. I wanted to see if PE5 is being ubiquitinated due to its interaction. So I again performed a directed co-IP, this time using flag tagged ubiquitin. And this is a typical ubiquitin blot where you see a laddering pattern that is seen in the GFP expressing cells. But this laddering pattern is not there in PE5 expressing cells, suggesting an absence of PE5 ubiquitination. And that's also quantified here. And this is strange that it interacts, but there's no ubiquitination. But I anyway wanted to check if there was a proteasomal degradation. So to test that, I used two inhibitors, MLN4924 from before, and also MG132, which is a proteasome inhibitor. And when I used these two inhibitors, I saw an increased protein expression of PE5 in the presence of these two inhibitors, suggesting that although there is no ubiquitination, there is still proteasomal degradation of PE5. Now, this was to answer this, we came up with this hypothesis that PE5 is probably dragging a non-CRL2 substrate, and this non-CRL2 substrate is maybe getting ubiquitinated, and PE5 is also degraded due to the complex that it forms with this ubiquitinated product. And while I was trying to find what is this substrate that is being degraded by PE5, I came across this really interesting phenotype where this membrane, a plasma membrane associated receptor, FPN1, in GFP expressing control cells, it's present in its natural position of plasma membrane. But when PE5 is expressed, this FPN1 is retained within, or it forms puncta, which are characteristic of endosomes. And this phenotype is reversed in PE5 delta GG mutant. Because of this phenotype, we think that PE5 is probably altering something in the endosomal recycling complex, and that's why the trafficking of this membrane receptor is being altered. So currently we are trying to find the mechanism of how this is happening, but I would like to sum up my talk by saying that we identified PE5 interacts with CRL2 complex, and we are very excited that we could identify a new host restriction factor or the host restriction functions of this CRL2 complex. And we are interested in seeing if any other PEPPs, because this is a large family, there might be several other PEPPs which are also targeting CRL2, either through KLHDC2 or through other substrate receptors. We are interested in finding if any other PEPPs are also targeting. We hypothesize currently that PE5 is dragging a non-CRL2 substrate for degradation, and I'm currently working to identify which protein is destabilized by PE5. With that, I would like to thank everyone in my lab, especially my mentor, Dr. Bell, and please stop by our posters today. We have 2 other people from our lab, and thank you all.
So this PE5 is excreted from Mycobacterium cells.
Yeah. Yes.
Through like
secretion system, type
secretion system.
Yes.
So when you put it into Salmonella, how do you have it secreted?
Oh, I did not put it in Salmonella. I expressed PE5 in macrophages, so it's already present in macrophages, and I infected these macrophages with Salmonella.
Okay. So no biosecurity risks that we need to worry about?
No.
Okay.
So I guess I want to ask a technical question. How do you ensure that your tag is not affecting the endosomal localization?
So I had controls, right? So I have GFP, I have GFP PE5, and I also had the GFP PE5 delta GG mutant. Considering the mutant is not showing the same phenotype as PE5, I think the FPN1 mislocalization, I think it's not because of the tag, but it's because of PE5. So yeah, the only controls.
Any other questions? Okay, if not, let's thank Bala and all of the speakers from this afternoon session.